This week in medical and vector entomology, 111 new studies sketch a world where the smallest animals still decide the fate of the largest public health campaigns. The dominant thread is resistance. In Suriname, Aedes aegypti populations delivered the country's first report of malathion resistance, alongside the knockdown resistance mutations V410L, V2016I, and F1334C. Across Italy, the Mosquirate network mapped KDR alleles in Aedes albopictus nationwide, while in Oyo State, Nigeria, researchers measured the intensity, not just the presence of pyrethroid resistance in Anopheles gambii. The message is consistent: our chemical toolbox is thinning and the insects are keeping score. So the field is looking beyond chemistry. A major review argues that insecticide resistance must now be read through microbiomes, machine learning, and emerging molecular technologies, not genetics alone. And genetics itself is being turned into a weapon with a tool called CRISPR. So what is it? Think of CRISPR as molecular find and replace. Scientists design a short piece of guide RNA to spell out a target sequence of DNA, and a protein called Cas9 carries that guide along the genome until it finds an exact match. When it does, Cas9 acts like a pair of molecular scissors and cuts both strands of the DNA at that precise spot. The cell then scrambles to repair the break, and in that repair, researchers can knock a gene out completely or paste in new instructions. That precision is what lets scientists switch off the exact genes that make a mosquito a vector. In practice, an engineered promoter system enabled high-efficiency CRISPR editing in the malaria vector Anopheles sinensis, and CRISPR knockout of the white gene landed in Culicoides sonorensis, the biting midge that carries blue tongue virus. Modelers, meanwhile, explored releasing Wolbachia-infected males in synergy with insecticides, biology and chemistry combined against transmission. The pathogens are not standing still. The CDC posted a Level 2 chikungunya notice for Costa Rica, and a real-time framework tried to infer chikungunya outbreak sizes in mainland France. A global assessment charted dengue seasonality from 1990 to 2024, while a provocative analysis reframed urban development itself as a driver of dengue burden. Aedes aegypti was confirmed for the first time in Buraydah city. Saudi Arabia arranged expansion in real time at the edges. The threats are re-emerging and moving. The New World screwworm is flagged as a re-emerging parasitic threat, with new work comparing trap designs to catch it. Migratory birds are ferrying Hyalomma ticks into northeastern Spain, carrying the spectre of Crimean Congo hemorrhagic fever. In Minnesota, climate and environmental data now predict tick abundance and Lyme cases 1 to 2 years in advance, and a broader warning lands on climate change and the growing threat of bunioviruses. From resistant mosquitoes to bird-borne ticks, this week's science says the same thing in many voices. Surveillance is the front line, and the front line is everywhere.